Hello, Biol005B students. My name is Ogenoma Ogenesume. I go by OG for short just because it's significantly easier. And I'm one of your SIs. Today we're going to be going over some of the topics that I thought were a little bit more challenging from lectures 2 and 3 from Dr. Redak. And with that being said, let's go ahead and get started. So the first thing we're going to start off with today are prokaryotes, parts of them. But first off, what is a prokaryote? So basically, prokaryotes are small unicellular organisms that can pretty much thrive anywhere, meaning that they are extremely biochemically diverse and that they can somehow continue to function and operate in almost any environment or any extreme that you can think of. So like I'm talking bottom of the ocean, inside a volcano, within our digestive systems, like they can thrive anywhere. So these guys can low-key do the most. They come in three different flavors or three different morphologies that you're going to really need to know. The first one is basically a sphere or a cosi. As cosi is like I think the cosi is like the official name for them. You can call it cockeye, cosi. I don't know. I've heard it pronounced all the different ways. I don't think it really matters. But basically, these are little spheres. Another one would be the rods or the bacilli. And then the last one would be basically the little spiral shaped ones. And those are the three major morphologies that you're going to see prokaryotes in. But moving on to the parts of the prokaryotes, first thing I'm going to go over is the fact that they have a cell wall. And the main function of their cell walls are to maintain structure. And so the whole point of their cell walls are to help provide like kind of protection. And then also during osmotic imbalances, it helps kind of maintain the structure of the organism. And then the two things I want to kind of talk about with cell walls is that this is one of the actual few times that he distinguished the difference between bacteria and archaea. So both bacteria and archaea are both classified under prokaryotes, but however, bacteria, their cell walls are actually made up of peptidoglycan, where however, archaea, their cell walls, if they do have them, are usually made up of like sugar and protein components that do not include peptidoglycan. So please remember that on the test or just moving forward, those two cell walls are not made up of the same things. The next thing I'm going to go over is the fimbriae and the capsule. Both of those, I like to kind of keep it simple. They are sticky stuff. So basically the fimbriae and the capsule, they're essentially appendages or an outside covering of the organism that helps it either stick to itself or stick to desired substrates. So they're kind of helpful in just like within its environment. It's just kind of a cool ability to have that helps its relative environmental fitness. He didn't really go too much into that, so don't really worry about it too much. Just know that they're sticky stuff. That's how I'm gonna like leave for the video. The next thing I'm gonna go over is the flagella. So for the flagella, they are basically for locomotion. Now, one thing I do want to point out is that the flagella here, these prokaryotes, are not the exact same thing as the flagella as you see in eukaryotes. First of all, their size is ridiculously different. They're not even close to the same size because, like we said, prokaryotes are really, really, really small unicellular organisms, and eukaryotes are pretty freaking big. So not at all the same thing, and they have different mechanisms of operation as well, but they do look the same, and they're called the same thing, which is a little bit confusing, but just know they're not the same. Moving on to their circular chromosomes. So basically they have circular and non-bound chromosomes in a very relatively small genome. And so I think that's part of a really big thing that makes prokaryotes is the fact that they do not have a bound nucleus. They do not have a membrane-bound nucleus or organelles with the exception of ribosomes. So that's part of what makes prokaryotes, not eukaryotes. Last but not least is this little sex pilus thingy right here. And that weird looking structure is basically kind of what it sounds like. I kind of think of it as like sort of a bacterial sex penis, although it's kind of weird. But I mean, I guess they have to conjugate somehow, but it helps facilitate conjugation. And so that little structure that sticks out of them helps kind of connect with other bacteria and help for horizontal gene transfer. All right, so that pretty much summarizes the basics of prokaryotes. But moving on, so the next experiment I'm going to go over is the one Redak was talking about that basically proved that bacterial colonies or the population themselves can evolve, and they can do so at a relatively fast rate. The experiment, the actual logistics of it, basically all that happened was they got a tube. And so within this tube, you have some media, like some nutrient-rich, high glucose solution, and you had the bacteria. And that bacteria was like let to thrive and grow and reproduce. They took a 1 to 100 dilution, so they took zero point 0.1 milliliters from this tube and they put it into another tube. However, this tube had low levels of glucose and it was a very, very crap media. And so basically in this media, the bacteria aren't able to grow or reproduce as well. And they're gonna have a much lower fitness rate here than they would have had over here. So with that being said, they continued to do serial dilutions. And then with that serial dilutions, they were actually just taking a little bit of the bacteria after having letting it grow for a whole day and letting it kind of like reach a certain capacity, and then taking it into another tube, a little bit of it, the same 0.1 milliliters from this tube, and taking it into another tube filled with the same crappy media. All of the continued dilutions all had the exact same media. They're not kind of progressively getting worse. It's just the same ones, just a fresh batch. That's all it is. That's all it is. So the whole point of the serial dilutions, I believe, is to kind of constantly provide it with a fresh source of glucose and kind of a fresh environment so it can continue to reproduce, even though it may not be ideal. At least you're not getting overcrowded and depleting it from all of its resources. And the more that it's able to reproduce, the more that you're able to hopefully continue to see more beneficial accumulated mutations. So that's essentially what happened. So as you can see here from the data, you could see that 5,000 generations down the line, and note that is not 5,000 dilutions, but it's actually just with one generation, meaning 1 hour. If you divide 5,000 by 24, you actually get about like 208. So 208 tubes down the line, they took the bacteria from there, they put it into another fresh batch of crap solution. They took the bacteria from here, the original, and they put it into also another fresh batch of crap solution, and they compared their relative fitness. We're talking about relative fitness. Like I said before, relative fitness is their ability to survive, their ability to thrive and reproduce in their given environment. And what they saw from the data was actually that the bacteria 5,000 generations down the line, or 208 days down the line, actually had a 50% increase, or 1.5 times the relative fitness compared to the original bacteria, meaning that they had evolved and adapted to their environment significantly so. Please, please, please remember, these are not individual mutations from an individual organism. We're talking about many, many generations. So the whole point of this, and the rest of the graph kind of shows the same as you continue moving down in evolutionary time. And so all this is basically proving is that over a relatively short time, but with many generations, bacterial colonies can increasingly become more fit for their environment, for a new crappy environment. They can basically, their biochemical diversity allows them to adapt to their environment. And then it also proves the ability of bacterial populations. Remember, we're talking about population genetics, not, or we're talking about populations evolution, not individual species. It allows them to adapt to their environment despite their relatively slow mutation rates. And this is all caused by rare and accumulated beneficial mutations. So yeah, that's that's a basic summary of this experiment. Moving on to the next thing, we're still talking about prokaryotes, but with almost all things in life, there are pros and there are cons. So I'll start with the cons because those ones are a little bit more well known. So the biggest one of them all that we kind of focused on would seem to be human diseases, and 50% of all human diseases seem to have originated from prokaryotic pathogens. They've killed a lot of us off in human history, a lot of plagues, a lot of bad stuff, and it's been difficult. We've came up with antibiotics, but another, negative aspect about prokaryotes is their ability to adapt. So they're also their ability to form antibiotic resistance. So over generations, if you treat it with antibiotics, but you don't kill all of them off, then some of them will gain antibiotic resistance, like I said, over generations. Next thing I'm going to go over is the difference between endotoxins and exotoxins. Prokaryotes can sometimes be one or the other or both. And so for endotoxins, this is when within the bacteria, the actual toxins are held within them. So while they are living within the host, let's say a human body, they're not actually giving much or significant damage to the host, but it isn't until that they actually get lysed where the toxins are able to be released from the bacteria. But on the other hand, when we're talking about exotoxins, these are bacteria that while they're alive continue to release the toxins and cause significant damage to the host during their actual stay in the host. But that's it for the cons. It's the major ones for the cons. Moving on to the pros. These are a little bit less well known, but they're actually essential for waste breakdown and recycling. So as we talked about, they can adapt. They're very biochemically diverse, so they can break down and survive just about anywhere. So given enough time, they'll break down just about anything and make it reusable back for our natural environment. The other big pro is that they're really helpful for human digestion and immunity. They play a really big role. They are in our gut flora. So you might be a little bit confused, like why do we have so much bacteria that like stays within us as human beings? But for those who don't know, we actually have good bacteria, we have bad bacteria. So although the bad bacteria can cause harmful pathogens, the good bacteria help just keep everything in balance and play major roles in our digestion. Another big thing is for fuel. So they actually are really essential for ethanol production. With that ethanol production, that's made for gasoline. We're not talking about the ethanol in our drinks. Those are different. Those are yeasts, which are not prokaryotes, as you mentioned in the lecture. And the last thing I'm going to go over for pros is the nitrogen fixation. So they play a huge role into what our atmosphere is today. And without their critical role in the formation of our atmosphere and usable nitrogen, and I believe they play a big role in oxygen as well, we would not have anywhere near the same life sustainability that we do. All right, and so moving on. So next topic we're going to talk about is endosymbiosis. And with endosymbiosis, it's basically the major theory of how we started off with a prokaryote and then throughout evolutionary time gave lineage to our first eukaryote and photosynthetic eukaryote. And so basically the theory behind this is we have a plasma membrane. So we have the prokaryote with the plasma membrane, and then within that plasma membrane, within this plasma membrane right here, we have these invaginations that basically come up and surround the nucleus. And so with the nucleus now membrane bound, now we have our first primitive eukaryote right here. With our first primitive eukaryote, we have an aerobic heterotrophic prokaryote, which basically is the pre-mitochondria that also gets engulfed by the cell. And that's what you see in primary endosymbiosis. So this is what you have for primary endosymbiosis. And then moving on, eventually, not like this same exact cell, because it's not like this same cell took up a mitochondria and then eventually took up a chloroplast, but no, over evolutionary time, throughout secondary endosymbiosis, basically a chloroplast or a photosynthetic prokaryote gets also engulfed by another eukaryote, and that's when you lead to secondary symbiosis, leading to ancestral photosynthetic eukaryotes. And yeah, that's the main idea with endosymbiosis. All right, so I'm going to finish off this video going over the 4 large clades of eukaryotes, and they are the Excavata, the SAR, the Archaeplastida, and the Uniconta. You're obviously not going to have to memorize every last little fact about this whole tree, but those 4 different clades you're going to know, and some key facts as well. I took the liberty of just writing down some of the major key points. I'm not going to go through every one of them, so please feel free to pause the video and then write down these major points. They're also in the lecture slides as well. But yeah, for Excavata, one of the key things that I think you should really go over is just the fact that they have whip-like flagellum. That's one of the defining characteristics of the Excavata, and they have many of them have photoreceptor eye spots. For SAR, they have the locomotion through the cilia, beating in synchronization. They have parasites that can cause malaria, and also they're responsible for a ton of photosynthesis. Like I said, please pause the video and write down these key facts because I think all of them are important. So the last two clades we're going to go over are the Archaeplastida and the Uniconta. So with Archaeplastida, basically you got all the plants. You got all land, you got all aquatic plants. All of these guys are all photosynthetic, and it includes red and green algae, but not brown algae. Lastly, we're going to end off with the Uniconta, which has slime molds, amoebas, fungi. But lastly, and most importantly to me, us. And yeah, that's pretty much all that I have for you guys today for this video. Thank you for sticking through with me all the way to the end. If you have any questions, please feel free to reach out to me or any of the other SIs. Our contact information should be posted, sent to you guys through email. We have our SI schedule for you guys right here. Please take a screenshot or take a picture of it so you can know where to, when and where to meet any of the SIs. We're going to be located on Zoom. But yeah, thank you so much and have a great rest of your day. Later.